The endocrine system, along with the nervous system, controls body activities. In the nervous system, neurons provide rapid cell-to-cell communication at a distance with a short-lasting response. Most endocrine cells communicate slowly at a distance with long-lasting responses. Endocrine production cells communicate with target cells in the body using chemical signals called hormones. Most endocrine hormones are circulating hormones. These chemical signals travel through the bloodstream, facilitating communication between a production cell and a target cell. They pass from the secretory cells that discharge them into interstitial fluid and then into the blood. Only target cells with the appropriate hormone receptor will respond to the hormone signal. There are generally two chemical types of circulating hormones. Hydrophobic hormones, like estrogen, are also known as steroids and are derived from cholesterol. These hormones tend to be lipid soluble. Hydrophilic hormones, like insulin, are based on amino acids, so they tend to be water soluble. The chemical category, hydrophobic or hydrophilic, determines the method by which the hormone affects the target cell. Hydrophobic hormones are bound to a carrier protein as they travel through the bloodstream. These steroid hormones easily diffuse through the phospholipid bilayer of the target cell's membrane and bind with receptors within the nucleus. This binding forms a hormone receptor complex that directly affects gene expression. Targeted DNA sequences are then transcribed into mRNA. This results in the synthesis of new proteins. The new proteins are often enzymes, which alter the target cell's activity. They cause cellular responses that are unique to that hormone. Everything from changing metabolic or contraction rates to the synthesis of other chemicals, including hormones, may be altered under the influence of a hydrophobic hormone. In contrast to hydrophobic hormones, hydrophilic hormones cannot pass through the target cell's membrane. These water-soluble hormones must bind to a receptor on the membrane. When a hydrophilic hormone binds to the hormone receptor, a G protein is activated. The activated G protein stimulates the membrane-bound enzyme adenylyl cyclase, which converts ATP into a second signal or messenger, known as cyclic AMP. Cyclic AMP acts to activate pre-existing cellular enzymes known as kinases. In a cascade of events, these enzymes either activate or inhibit other cellular proteins. While the action of the second messenger, cyclic AMP, is short-lived, the net result of the hormone signaling is an alteration of the target cell's physiological response. Even though the mechanism of action is different in water-soluble hormones, the range of physiological responses can be similar to those found in steroid hormones. The homeostasis of many of our body's controlled conditions, such as blood pressure, blood glucose levels, or metabolism, are maintained by the mechanisms of action of the hormones that the endocrine glands produce. These chemical signals are vital to our well-being.
Endocrine cells, located in various parts of the body, produce a large variety of chemical messengers in the form of hormones. Once they are released from the endocrine cells, the hormones diffuse into surrounding blood vessels and are transported in the blood. The hormones are bound for their target cells, which may be some distance away from the point of secretion. Once they reach the site of the target cells, the hormones leave the bloodstream and attach to the target cell. Hormones can only affect their target cells when they are bound to specific target cell receptors. The manner in which the hormone interacts with the target cell depends on the chemical nature of the hormone. Based on their chemical structure and lipid solubility, Hormones are classified into two groups. Lipid-soluble hormones include steroid hormones and thyroid hormones. Lipid-insoluble hormones include amines, peptides, and protein hormones. Lipid-soluble hormones, such as steroids and thyroid hormones, can easily pass through the lipid-rich plasma membrane. Once inside the cell, the hormone binds with an intracellular receptor, which is either in the cytoplasm or the nucleus of the target cell. This results in the formation of a hormone receptor complex. The hormone receptor complex is transported into the nucleus. The hormone receptor complex influences gene activity, resulting in the DNA transcription and the production of messenger RNA. The mRNA leaves the nucleus and is translated to form a new protein in the cytosol. The new proteins alter the cell's activity. Lipid-insoluble hormones, such as peptide hormones, cannot pass through the plasma membrane and enter the target cell. They can only interact with the surface of the target cell. These hormones bind to membrane receptors on the outer surface of the target cell's plasma membrane. The binding of the hormone to its receptor sets off a sequence of events. First, it activates a G protein found on the inner surface of the membrane. In turn, the activated G protein transforms the signal from the receptor and activates the amplifier enzyme adenylate cyclase. The activated adenylate cyclase converts a large number of ATP molecules to cyclic AMP molecules, thus amplifying the signal received from the first messenger. Cyclic AMP works as the second messenger inside the cell. It activates kinase proteins. Kinase proteins activate other enzymes through phosphorylation reactions. Phosphorylated enzymes can trigger a host of biochemical reactions that have profound effects on the target cell's activity. In this way, lipid-insoluble hormones, which cannot enter the cell, can still influence cell function through a second messenger, cyclic AMP, inside the cell. The endocrine system regulates many body conditions with feedback loops. Each feedback loop has the following components: stimulus, a change in a body condition; production cell, an endocrine cell that produces a hormone after being affected by stimulus; hormone, the signaling chemical; target cell, a cell receptive to the hormone; action, what the cell does when affected by the hormone; response, the overall change in controlled body condition as a result of the feedback loop. To learn more about specific hormone effects on various controlled conditions, follow these directions. Select the controlled condition from the menu bar above the diagram. Then, choose how you would like to stimulate that condition. Finally, choose a hormone from the list provided. To explore all the hormones and their effects on the controlled conditions, always begin by choosing a controlled condition first. The endocrine system regulates many body conditions with feedback loops. Each feedback loop has the following components: stimulus, a change in a body condition; production cell, an endocrine cell that produces a hormone after being affected by stimulus; hormone, the signaling chemical; target cell, a cell receptive to the hormone; action, what the cell does when affected by the hormone; response, the overall change in controlled body condition as a result of the feedback loop. To learn more about specific hormone effects on various controlled conditions, follow these directions. select the controlled condition from the menu bar above the diagram. Then, choose how you would like to stimulate that condition. Finally, choose a hormone from the list provided. To explore all the hormones and their effects on the controlled conditions, always begin by choosing a controlled condition first. Exercise, or not having recently eaten, causes a decline in blood glucose concentration that stimulates alpha cells in the pancreatic islets to secrete glucagon. Glucagon targets liver cells and causes them to undergo glycogenolysis, a process that breaks down glycogen into glucose. It also promotes the formation of glucose from non-carbohydrate sources, a process called gluconeogenesis. The glucose is released into the blood, and blood glucose concentration is restored to normal levels. A decline in blood glucose concentration caused by increased metabolic needs or physical exertion stimulates the release of growth hormone-releasing hormone from the hypothalamus. The releasing hormone targets the somatotroph cells in the anterior pituitary to secrete human growth hormone. Human growth hormone targets liver cells and causes them to undergo glycogenolysis, a process that breaks down glycogen into glucose. The glucose is released into the blood, and blood glucose concentration is restored to normal levels. Human growth hormone also targets adipose cells. HGH causes adipose cells to undergo lipolysis, a process which allows cells to use fats for energy instead of glucose. With cells using fat for fuel, blood glucose is spared, so blood glucose concentration is returned to normal levels. In summary, HGH causes liver cells to undergo glycogenolysis. HGH causes adipose cells to undergo lipolysis. The resulting response of HGH secretion to both cell types is the increase in blood glucose levels. A decline in blood glucose concentration stimulates corticotropic cells in the anterior pituitary to produce adrenocorticotropic hormone, ACTH. ACTH binds with cells in the zona fasciculata of the adrenal cortex and promotes the production of cortisol, the major glucocorticoid in the adrenal cortex. Cortisol targets liver cells and causes them to undergo glycogenolysis, a process that breaks down glycogen into glucose. It also promotes the formation of glucose from non-carbohydrate sources, a process called gluconeogenesis. The glucose is released into the blood, and blood glucose concentration is restored to normal levels. Cortisol targets muscle cells and causes them to break down protein, thereby liberating amino acids into the bloodstream. The amino acids are used by the liver to produce more glucose. The glucose is released into the blood, and blood glucose concentration is restored to normal levels. Cortisol also targets adipose cells. It causes adipose cells to break down lipids, a process called lipolysis, thereby liberating fatty acids into the bloodstream. The fatty acids are used by the liver to produce more glucose. The fatty acids are also used by other cells for fuel, thereby sparing glucose and allowing the return to normal blood glucose levels. The glucose is released into the blood, and blood glucose concentration is restored to normal levels. In summary, ACTH promotes production of cortisol. Cortisol causes liver cells to undergo gluconeogenesis. Cortisol causes muscle cells to break down proteins to amino acids. Cortisol causes adipose cells to undergo lipolysis. The total response of the actions of cortisol is to increase blood glucose levels. Certain situations, such as the absorption of a meal in the digestive tract, will cause blood glucose levels to rise. A rise in blood glucose levels stimulates beta cells in the pancreatic islets to secrete insulin. Insulin can bind to many cells in the body. Under the influence of insulin, the body cells increase uptake of glucose and amino acids. As glucose is removed from the blood glucose concentration is restored to normal levels. Insulin binds to liver cells. Under the influence of insulin, liver cells convert glucose to glycogen, a process known as glycogenesis. As glucose is removed from the blood glucose concentration is restored to normal levels. Insulin can bind to adipose cells. Under the influence of insulin, adipose cells convert glucose into triglyceride and fats, a process known as lipogenesis. Blood glucose declines towards normal levels. Increased insulin causes increased transport of glucose out of the blood, formation of glycogen, formation of fat. The total response of all the actions of insulin is to decrease blood glucose levels. Cellular needs for calcium may cause blood calcium concentrations to decline below the normal range. This decrease stimulates the principal cells in the parathyroid gland to secrete parathyroid hormone. Parathyroid hormone binds to osteoclasts in bone tissue, promoting bone resorption and the release of calcium into the blood. As calcium enters the blood calcium concentration is restored to normal levels. Parathyroid hormone also binds to kidney cells and promotes the production of calcitriol. Calcitriol binds with intestinal cells, causing them to increase absorption of calcium, which enters the bloodstream, restoring calcium levels to normal. Increased parathyroid hormone secretion promotes bone resorption and the production of calcitriol. The result of these actions is an increase in blood calcium levels. Nutrient absorption in the small intestine may cause blood calcium concentrations to increase. This rise stimulates parafollicular cells in the thyroid gland to secrete calcitonin. Calcitonin interacts with osteoblasts in bone tissue and stimulates them to take up blood calcium and deposit it in the bone matrix. This lowers the blood calcium concentration to normal. Dehydration, blood loss, and low amounts of water in the blood can cause blood volume and pressure to decrease. In response to declining blood volume or elevated blood osmotic pressure, neurosecretory cells in the posterior pituitary release antidiuretic hormone. In high concentrations, ADH can also cause vasoconstriction in the walls of arterioles. The narrowing of the lumen in these blood vessels raises blood pressure. ADH binds to the principal cells, forming the walls of nephron tubules in the kidney, stimulating them to synthesize water pores, and thereby increasing the permeability of the tubules to water. The reabsorption of water reduces water loss from the body, increasing blood volume and returning blood osmotic pressure to normal. ADH binds to the sweat glands. The secretory activity of the sweat glands decreases, lowering the rate of water loss through perspiration. Increased release of ADH causes water reabsorption in the nephron tubules of the kidneys, vasoconstriction of smooth muscle in arterioles, inhibition of sweat gland secretion. The total result of these actions is an increase in blood volume and pressure. In response to declining blood volume and pressure, juxtaglomerular cells of the nephron in the kidney release renin into the blood. Acting as a catalyst, renin aids in the conversion of angiotensinogen to the hormone angiotensin II. Angiotensin II targets smooth muscle cells in arterioles that provide blood to the nephron and causes these blood vessels to constrict. Because less blood is filtered by the nephron, less water is lost in urine, so blood volume and pressure rises. Angiotensin II targets the zona glomerulosa cells of the adrenal cortex and promotes production of another hormone, aldosterone. Aldosterone circulates in the blood to the kidneys. There, it promotes the reabsorption of ions, particularly sodium, from urine, and indirectly, water, back into the bloodstream. These actions result in raising blood volume and pressure. Angiotensin II targets cells in the proximal convoluted tubule of the nephron within the kidney. Angiotensin II promotes the reabsorption of sodium and chlorine ions, setting up an osmotic gradient favoring the retention of water. Less water is lost in urine, so blood volume and pressure rises. In summary, renin promotes the production of angiotensin II. Angiotensin II causes vasoconstriction of arterioles. Angiotensin II promotes production of aldosterone. Aldosterone promotes reabsorption of ions and water. These combined activities result in increased blood volume and pressure. Many situations can elevate blood volume and pressure. In response to rising blood pressure, atrial cells in the heart will stretch, stimulating the secretion of atrial natriuretic peptide. ANP targets muscle cells in blood vessels and causes them to relax. This results in vasodilation and the lowering of blood pressure. Atrial natriuretic peptide also targets cells of the proximal convoluted tubule in the nephron of kidneys. There, it inhibits the retention of sodium ions, which reduces the retention of water. Less water returning to the blood causes blood pressure to return to normal. High levels of angiotensin II result from low blood pressure or low levels of sodium ions or high potassium ion blood concentration. These high levels stimulate the production of aldosterone from the zona glomerulosa cells of the adrenal cortex. Aldosterone binds to the principal cells of the collecting ducts of the nephron, where it promotes increased sodium, chlorine, and hydrogen carbonate reabsorption from urine, increased water retention and reabsorption back into the bloodstream, and increased potassium and hydrogen secretion. The conservation of sodium, chlorine, and hydrogen carbonate ions, as well as water, and the elimination of excessive potassium and hydrogen ions, results in an increase in blood volume and pressure. In response to a decreased metabolic rate or blood glucose concentration caused by increased metabolic need or physical exertion, the hypothalamus increases secretion of thyrotropin hormone-releasing hormone, or TRH. Thyrotrophic cells in the anterior pituitary respond to rising levels of TRH and secrete thyroid-stimulating hormone, TSH. Thyroid-stimulating hormone binds to follicular cells of the thyroid gland and stimulates them to secrete thyroid hormones T3 and T4. Thyroid hormones bind to body cells, causing increased oxygen use and basal metabolic rate, an increase in cellular metabolism due to the production of more ATP from increased glucose catabolism, an increase in lipolysis, providing cells with energy for cellular metabolism from lipids. The overall result of these actions is increased fuel for a faster metabolic rate. The increased metabolism will result in increased body temperature. The thyroid hormones target muscle cells in blood vessels and the heart. The hormones cause vasoconstriction of blood vessels and contraction of the heart muscle, both of which increase blood pressure. The increased blood pressure and flow enhances the delivery of fuel molecules and oxygen to cells, supporting a higher metabolic rate. Thyroid hormones target body cells, including the skeletal, muscular, and especially nervous system cells during growth and development. Increased thyroid hormones increase protein synthesis, which promotes a metabolic rate necessary for normal growth and development of skeletal, muscular, and nervous systems. Low blood glucose, physical exertion, and increased sympathetic stimulation trigger the release of human growth-releasing hormone from the hypothalamus. In turn, HGRH stimulates the production of human growth hormone from the somatotrophic cells of the anterior pituitary. Human growth hormone targets cartilage, bone, and skeletal muscle cells. In these cells, HGH increases protein synthesis, increases mitotic rate, and increases the levels of fuel molecules, glucose and fatty acids, in the blood, which supports increased cell growth. The overall response to HGH is increased cell numbers, supporting normal growth and development of the skeletal and muscular systems. Stressors, such as declining blood pressure due to blood loss or strong emotional reactions, trigger increased sympathetic stimulation from the hypothalamus to the chromaffin cells of the adrenal medulla, causing the immediate release of epinephrine and norepinephrine. Epinephrine and norepinephrine binds to the cardiac muscle cells of the heart. These cells constrict faster, increasing the heart rate and therefore cardiac output. Increased cardiac output rapidly increases blood pressure, thereby resisting stress. Epinephrine and norepinephrine also binds to smooth muscle cells of blood vessels. This alters the flow of blood through the vessels by increasing the flow through the brain, heart, lungs, and skeletal muscles, and reducing the flow of blood through the skin, urinary, digestive, and reproductive organs. Blood flow is diverted to those organs that are capable of rapidly resisting stress. Epinephrine and norepinephrine binds to liver cells. The hormones increase blood glucose by promoting glycogenolysis. Increased glucose is delivered to those organs that are capable of rapidly resisting stress. Epinephrine and norepinephrine binds to the smooth muscle cells in bronchioles. This promotes bronchodilation, allowing for greater airflow into the lungs. As a result, increased oxygen is delivered to those organs that are capable of rapidly resisting stress. In summary, epinephrine and norepinephrine promotes increased cardiac output, altered flow of blood through the vessels to appropriate organs, glycogenolysis, and bronchodilation. The overall result of these actions is an enhanced ability of organs to resist stress. Stressors such as declining blood pressure due to blood loss or strong emotional reactions stimulate the production of a hypothalamic releasing hormone, cortisol releasing hormone, CRH, as well as adrenocorticotropic hormone, ACTH. In turn, these hormones stimulate the production of the hormone cortisol in the zona fasciculate cells of the adrenal cortex. Cortisol binds to muscle and liver cells. It causes muscle cells to break down protein and liberate amino acids into the bloodstream. The liver uses the amino acids to produce more glucose. Increased glucose is delivered to cells and organs so stress can be resisted. Cortisol binds to smooth muscle in blood vessels. It causes vasoconstriction, which counteracts a drop in blood pressure due to blood loss. Blood flow is maintained during stress. Cortisol binds to macrophages and lymphocytes. An increase in cortisol inhibits a macrophage's production of inflammatory producing chemicals, which helps reduce swelling in tissues. In addition, it inhibits proliferation of lymphocytes, reducing the immune response. In summary, cortisol promotes the breakdown of proteins for increased gluconeogenesis, vasoconstriction to raise blood pressure and blood flow, and suppression of immune responses. The overall result of these actions is greater ability of the body to resist stress. Stressors, such as declining blood pressure due to blood loss or strong emotional reactions, stimulate production of hypothalamic releasing hormone, GHRH. GHRH stimulates the somatotroph cells of the anterior pituitary to secrete human growth hormone. HGH causes liver cells to undergo glycogenolysis, the process that breaks down glycogen into glucose. In addition, HGH causes adipose cells to undergo lipolysis, which mobilizes stored lipids for energy use. The increased nutrients are released into the blood, travel to body cells, and are used to increase available ATP within the target cells. Stressors, such as declining blood pressure due to blood loss or strong emotional reactions, stimulate production of hypothalamic releasing hormone, TRH, which stimulates the production of thyroid stimulating hormone, which targets the follicular cells of the thyroid gland to produce thyroid hormones. The thyroid hormones target body cells in order to promote an increase in cellular metabolism due to the production of more ATP from increased glucose catabolism, and an increase in lipolysis, providing cells with energy for cellular metabolism from lipids. The overall result is an increase in oxygen, lipid, and glucose used to fuel a faster production of ATP within cells in order to reduce stress. Stressors, such as declining blood pressure due to blood loss or strong emotional reactions, stimulate production of hypothalamic releasing hormone, cortisol releasing hormone, and adrenocorticotropic hormone, ACTH, which in turn stimulate the production of aldosterone from the zona glomerulosa cells of the adrenal cortex. Aldosterone binds to the cells of the distal convoluted tube and collecting ducts of the nephron, where it promotes increased sodium, chlorine, and hydrogen carbonate reabsorption from urine, increased water retention and reabsorption back into the bloodstream, and increased potassium and hydrogen secretion. The conservation of sodium and elimination of hydrogen prevents a lowered body pH during stress. In addition, water is conserved due to the osmotic gradient produced by sodium retention, which helps maintain blood volume in case of blood loss.